your PhD might take 10 years, right? So, but of course, we work with them as well to try to crack this. So we wonder, how can you do this, apart from this counting game, okay? So we're thinking, well, one way to do that is to say if the circulating fetal DNA and mother's DNA have any molecular differences. And one very fundamental molecular difference is size. And there are many ways you can measure the size of molecules. Of course, some of you in the audience will use mass spectrometer, some will use electrophoresis. But here, if we use sequencing, we can also do that. So let's say this is a DNA molecule. Instead of sequencing one end of a molecule, let's say we sequence both ends. And then we know the human genome sequence. I can use a computer to map both of those ends back to the genome. And then by subtracting one coordinates from another one, I've got the size. So when we first start this project, I would imagine, like many biological parameters like height or weight, it will follow some sort of normal distribution. So I was very surprised when we did experiment. We find the distribution is anything but normal. So this is a size from 50 base pair to 200 base pair, and this is a frequency of the molecules. And then when we do that, we can see actually the distribution seems to follow a series of peaks. There's one peculiar peak at 166 base pair, and after that, every 10 base pair, there's another peak. When I first got the data, I thought it's some sort of artifact. And then, when you look at the fetal DNA, which is in blue, the distribution is on the left of the maternal DNA. So indicating maybe fetal DNA is shorter than mother's DNA. So what does all that peaks mean? So to know what all that peaks mean, we have to remind ourselves how the DNA is actually packaged into a chromosome. Because the DNA in your genome, each cell actually contains DNA which is about 2 meters long. So imagine how can you package 2 meters of like a shoestring into the small nucleus of a cell. And the answer is that the DNA will coat around proteins called histones and then gradually package into chromosome. So the most interesting thing is over here, when you can see actually the DNA will coat around twice in this histone molecule, and then there's an area called linker, and then go into the next one. And each of those units, like beads, is called a nucleosome. As it turns out, this bit is 146 base pair, this bit is 20, and the two together is 166. So what we're proposing is actually this bit is the core bit plus the linker, and this bit is just a core without a linker. And this is the difference between fetal and maternal DNA. So the fetal DNA, for some reason, actually is lacking this linker, so it's shorter. And after that, the 10 base pair peaks has to do with the fact that DNA is a double helix. And every 360-degree turn of double helix is 10 base pair. So this bit is actually telling you some secrets about how the DNA is being digested in plasma. So now, if this model is correct, if you can find me one species of DNA which doesn't have this nucleosome structure, then it will not have those peaks. And interestingly, we can actually find that in the form of mitochondrial DNA. So mitochondria are the powerhouse of cells, like batteries of cells. And strangely, mitochondria also have its own genome, its own DNA. And evolutionarily speaking, mitochondria actually come from some sort of bacteria. And bacteria don't have this nucleosomal structure. So you can see when you look at mitochondrial DNA plasma, it doesn't have those peaks. Actually, it's very unstable, it's very short, because the protein actually protects it. So now, with the elucidation of all that about size, we think that we are able to construct a new type of testing which is based on size. Then you say, how do you do that? The crux of that is, if the fetal DNA is shorter than maternal DNA, then the more fetal DNA you have in a particular sample, the shorter it should be. Now, for example, here you have two samples. One have 10% fetal DNA, one have 20%. So the 20% one will have more short molecules. So you look at the short guys, then the 20% one has got the high peaks than those with 10%. But on the other hand, if you look at the longer molecules on the right-hand side, then the more fetal DNA there is, the less of those long guys you have got. So then, of course, every time you have to look at a graph like that, it is pretty complicated for a simple doctor like me. So we decided to actually do a simple measurement called the size ratio, which is basically very easy. It's just a ratio of the short molecules over the long molecules. So the bigger is the size ratio, the shorter it is. And interestingly, when we measure the size ratio, we find actually it is proportional to how much fetal DNA you have in terms of the Y chromosome concentration. But on the other hand, this size ratio is better because the DNA method can only be used if the baby is a boy. But this method, you can use it for all pregnancy, boys and girls. And then we wonder, can we push this technology even further? So what I mean is this. So imagine in the mother's blood, there's some mother's DNA which is long, and there's some fetal DNA which is short. And normally you mix them together and give you that size ratio. So now imagine if you're looking at chromosome 21, and if the baby have Down syndrome, you have an extra dose of this short DNA. So theoretically, this extra short DNA should drag down the size in chromosome 21. Okay, so then we actually measure the size in different chromosomes. For example, measure chromosome 21. Then can find that the trisomy 21 pregnancy are different from the other cases. But on the other hand, if you look at chromosome 18, then those people with free chromosome 18 are different. On the other hand, if you look at chromosome 13, then those people with free chromosomes are different. So now you have a new way, which is based on size, which is different from the counting method. So we believe actually this counting and the size method are synergistic with each other. And actually, you can actually combining them, but each of those is 99.5% accuracy. And now by combining those, I think you can even push it towards a diagnostic test. Actually, this technology we have actually recently licensed to Illumina, and actually they are going to incorporate this into a